Hello, my name is Rita Ankumah, and the disease I did for my project is cholera. Cholera is a severe diarrhea disease that occurs when the intestine gets infected by the toxigenic bacterium Vibrio cholerae serogroup 01 or 0139. The disease remains in a serious public health threat, indicating inequality and lack of social development. The only species causing cholera is Vibrio cholerae. However, different serotypes of the pathogen exist, with the main types being Vibrio cholera 01 and Vibrio cholera 0139. The Vibrio cholera 01 is responsible for the majority of cholera cases worldwide, and it contains two biotypes called classical and L12. The Vibrio cholera 0139 is relatively new, as it emerged in the 1990s, being the major cause of the disease in Asia. However, it has similar clinical features to the 01 serogroup. Signs and symptoms of cholera are diarrhea, vomiting, leading to dehydration, and muscle cramping, low blood pressure, rapid heart rate, and electrolyte imbalances. Cholera is caused by a gram-negative curved rod-shaped bacterium, Vibrio cholerae. It produces cholera toxin and enterotoxin, causing excessive water secretion and electrolyte into intestines. V. cholera form biofilms and colonize copepods and other aquatic plants, helping in their survival. The pathogen survives in brackish salty water and can be found in untreated sewage and other contaminated water sources. This is the structure of Vibrio cholera. As one ingests Vibrio cholerae in contaminated food and drink, it will break the acidic barrier in the stomach, which causes production of cholera toxins. This toxin binds to G protein coupled receptor, leading to activation of adenylate cyclase, which inhibition of sodium and chloride intestinal absorption. This will lead to increased water and electrolyte secretion, which will cause severe dehydration and electrolyte imbalance, causing the transmission and the spread of cholera. Virulence factors. Virulence factors enhances the bacterium's ability to colonize the intestines, adhere to epithelial cells, and lead to severe disease signs and symptoms. Some key virulence factors include cholera toxin, which bind to specific receptors and disrupt normal cellular processes. Mucinase. This is an enzyme which degrades the protective intestinal mucus layer for the Vibrio cholera to access the underlying cells and start infections. Co-regulated pilus helps the pathogen during adhesion to the intestinal epithelial cells, which the hemagglutinin bacterium produces, which clamps red blood cells and cleave proteins. Helps the pathogen adhere to the intestinal epithelium and lead to tissue damage. Transmission of cholera. The disease is mainly transmitted when a healthy person ingests food and water contaminated with bacterium. Cholera also spreads via contaminated water sources. Vibrio cholera is common in raw undercooked food like, shellfish. Also, fruits and vegetables irrigated with contaminated water can transmit the bacteria. Person to person, though it's rare, direct transmission can also occur. Epidemiology. Around 1.3 to 4 million globally get cholera yearly, while about 21,000 to 143,000 people die from it. Cholera is most common in developing and poor countries of Africa, Asia, and Latin America. It was common in the 1800s because of the unhygienic practices. This bacterium can be detected by dark field or phase contrast tool microscopy. Selective culture media is often the initial step since it supports Vibrio bacteria growth and inhibits others' growth. Bacteria on TCBS agar are always yellow, round, and may have slight translucent or greenish blue color, as you can see. Additional tests are conducted to confirm the presence of the bacterium, and they include biochemical tests and molecular techniques like PCR. Based on the signs and symptoms, rapid diagnostic tests detect cholera toxins. Serological tests are vital but not common because of time constraints. Laboratory tests like the stool sample analysis using fresh stool with the following tests are performed. Polymerase chain reaction helps detect genes and DNA sequences to the bacterium. Culture on TCBS agar plates to identify bacterial features and appearances. Direct microscopy reveals comma-shaped or S-shaped pathogen. Treatments. Rehydration therapy, that is restoring fluids and salt loss during diarrhea and vomiting. Also, antibiotic therapy helps shorten diarrhea and reduce severe symptoms. Common antibiotics that are used in treating cholera include tetracycline, doxycycline, zithromycin, and ciprofloxacin. Zinc supplements are also good, very vital, treating cholera in children. And as you can see, these are the dosage for all these different antibiotics. Tetracycline capsule is 250 milligrams, 2 caps 4 times a day for 3 days. Children, the dosage in children will reduce. Erythromycin tablet, 250 milligrams, 2 tablets 4 times a day for 3 days, and also erythromycin in children will be 12.5, half of the dose of adults. These are some of preventative measures for cholera. Even though treating the disease is not complicated, it has a high mortality rate and kills in a short time. Preventing this disease, preventing the disease include enhancing safe water supply by ensuring that water is treated even from the source and it meets quality standards, high standard of sanitation and hygiene. That is by improving hand hygiene, safe food handling, more healthy education, and adding more sanitation facilities in the communities. Vaccines are also helpful, and some oral cholera vaccines such as Dukoral and Chancro vaccines have helped reduce the disease. These are all my references.